Hello, DEAF1 community. My name is Allyson Muotri. I'm a faculty, a professor here at UC San Diego in pediatrics and cellular molecular medicine. Pedro from the DEAF1 community has contacted me. He has a child with DEAF1 mutation, and he heard about our program here at UCSD, and he has asked me to introduce myself, introduce the research in my lab, and explain more or less what we do. So I'll try to do that real quick, and I'll be happy to have another meeting with the community to solve any potential questions or concerns that you guys might have. I'm sharing my slides here. So my lab has been focusing on modeling autism using human brain organoids. I'll tell you in a moment what is human brain organoids, and my motivation is it's right here. So this is Ivan, my son, who is on the spectrum. We also have a mutation, a genetic mutation. So the problem with those genetic mutations in the spectrum of autism is that they all occur very early on in life and affect neurodevelopment. And we scientists, we don't have access to in utero development, so we cannot actually study the human brain while it's developing. So all the process that leads to a developmental delay, seizure, things like that, a little bit late in life actually happens in utero, but we don't have access. So most of the biomedical community has been rely on different models to try to understand what's going on inside the human womb or uterus. We take advantage of postmortem tissues from patients that are deceased. Postmortem tissues, it's useful, but these are not live cells. We cannot study synaptogenesis. We cannot see how the brain is formed. The damage is already there. We already know that the patient who passed away have a problem. And we want to catch early. We want to understand early on how the mutation on the F1 gene can actually lead to neurodevelopmental disorders and the behaviors that we see in our kids. We also studied the blood of these patients, but blood cells are not brain cells. Blood cells do not form synapses. Blood cells do not form a brain. Blood cells does not tell you how the human brain behaves. So it's very useful from the genetic perspective, but it's the wrong model to understand the human brain. And finally, most of the biomedical research has been focusing on the creation of mouse models. So you use techniques to introduce mutation in the target gene, in DEAF1 gene, and then we look for phenotypes in these animals and trying to mimic the disease. There's a huge problem with mouse models, and I think all my colleagues will agree, is that once we start the clinical trials using the information that we have with these mouse models, and that takes several years from the basic preclinical discovery to initiate a clinical trial. 99% of these genetic alterations and the interventions that we found in mouse models do not translate into treatments for humans. So that's why the industry has basically given up on treating neurological conditions, especially neurodevelopmental conditions, because we don't have good models to do that. So, I mean, the last blockbuster in drug for the nervous system has been in the 70s, Prozac, and was actually found by chance. So the lack of models really inhibit the research to go forward. It's not that those models are not useful, they are just not translatable. The mouse is not a small human, so it does not translate what we learn. So it's great for basic science. It's terrible for translational research. So more recently, the biomedical community has been focused on brain organoids. So these are neurotissues that can be made from stem cells. This is one of the original publications showing that you can guide these cells to actually form these neurostructures. So they are actually here mimicking how the human fetal brain behaves. So this is great because now we have a model of the human fetal brain outside the uterus in a control condition of the lab. So my lab and others are specialized on creating tissues using these stem cells. The good news is that these stem cells can be derived from each one of you. So it can take like a piece of skin or a drop of blood, and reprogram the blood cells or the skin cells into what we call induced pluripotent stem cells. And from these cells, we can make these brain organoids. And we have been doing this for a while. There are limitations in the technique. These brain organoids are not mini brains. They are not miniaturized version of the brains. They are tissues that mimics how the brain behaves. So these are some of the limitations that over the time we're still increasing our protocols to better mimic the brain. The progress in this arena has been tremendous. This is a postdoc in the lab actually growing these brain organoids. You can actually see these white dots in there. So these are thousands of brain organoids that we produce from a child with autism. So it's not that difficult. It's quite easy to do. The cells self-organize by themselves. You don't have to guide them. What you are seeing here is a two-dimensional representation of how the organoid that form the sphere is from the progenitor cells in the center of the organoids that migrates to the outside and form this sphere, form the cortical plate. And the cortex has been like a structure that has implicated in autism, in seizures, things like that. That's why we like to focus on the cortex, but in theory, we can make different brain regions. This is a slice of one of these organoids. You see the cells self-organize very nicely, exactly as they do in the human brain. And we can even place these cells on electrodes so we can capture their electrical activity, and we can, over time, record from these organoids to see how they form these networks. And they actually form very complex networks, such as neural oscillations. These are oscillations that you can detect by EEG. So we can actually measure these oscillations in the organoids and now compare to the EEG of the same person where the organoid were derived from. So this technique has tremendous applications. This is one article of the New York Times talking about our research, just showing the potential for these organoids to actually mimic the human brain. And of course, there are some ethical implications here that I will not even discuss. But nonetheless, it's a great model for neurological and neurodevelopmental disorders. So we are actually applying this model to find treatments, both pharmacological treatment as well as gene therapy. So this is just a list of the diseases that I have going on in my lab. So we recruit patients, we reprogram cells from these patients, we create these brain organoids. If we know the gene, that's the case of DEAF1, we try to do a gene therapy or correction using CRISPRs. We can also try to use repurposed drugs to move on into clinical trials. And we've done that several times already. So we know that the model is useful. So we can enter clinical trial really fast by screening drugs that are already FDA approved. So that's one potential application. The other one, as I mentioned, gene therapy. This is just one example of gene therapy for Rett syndrome. So we hope this technology would be in clinics next year. It was supposed to be this year, but the company couldn't do it because of the epidemic. But here, what we are seeing is we're placing these organoids on the multi-electrode array, so we can measure the electrical activity right here. And in green, it's inside of the organoids where we inject the virus to deliver the correct gene inside the mutant cells. And by doing that, we were able to restore or rescue the electrical activity. So here is just, we are increasing the virus in Rett syndrome organoids. And in this condition already, which targets about 10% of the cells, we already see a significant amelioration of the activity of these networks. So very promising technology. I mean, gene therapy is now already in use for several disorders. So we hope neurodevelopmental disorders is a good target because you can target early on while the patient is a child and let them develop. So most likely the trajectory would be different with a treatment like that. So this is another condition where we see the electrical activity of the organoids going up. This is a condition that has early seizures. So by gene therapy, we can reduce the hyperexcitability of the neurons inside the organoids. And our hope is also to move this one into clinics to reduce the seizures. So these are just two nice examples. So these are people in my lab. I have funding from the California Regenerative Medicine Agents, as well as NIMH. And as I mentioned, we have several programs with different genetic mutations creating these organoids and really driving them towards clinical trial. So that's in short what we do here in the lab. I'm happy to discuss this further. Thank you so much.